So we have this. Now the next is for us to cut through the second layer. Now is the second layer that is going to be taking us straight into the internal anatomy. So again, we'll make a cut because you have to be very careful. It's another layer. So we have to make a cut through the layer so that we don't cut through the other organs. So I have my cut. Can you see it?
Yeah.
And then I cut through it again. Can you see it?
Yeah.
Now I am at the chest region, so I will need a stronger scissors. So this is the next one. You can see. Now if you're using this, the blunt end should be inside so that you don't cut through any organ, while the sharp end should be outside. So I will use this to cut through the, can you see it? I'm cutting through the ribs. Can you see it? Can you see it? And each time we do that, we try to watch out for blood. So we are trying to open the chest region. Now, at this point, we can actually look at what we have before we proceed. Okay? So we have this layer, I'll have that layer. We can equally also clamp this so that we can have clarity, be able to see deep, clearly. Okay. Now, as students, you are expected to have what is known as a chart, okay, that shows this procedure, so that the moment you open up, you compare with the chart. I think other students subsequently may have this chart to be able to compare. So at this point, you are having this. So I think we need to still open it up. All right, so this is what we have got. I think we need to pin this so that it will allow us to see. Okay. All right, so right here now, you can see we have opened this up, and if you check, you see the heart beating. Yes. Have you noticed that?
Yes.
That means it's not completely dead. And then you can study you can see how your heart beats. Can you see it? This is one thing that you must learn from here. You see? If we are actually doing surgery for anything here that will require us to still close up the animal and make the animal survive, this is the point at which we can do that. So the heart is beating even though it is completely lying down lifeless. So let's quickly go through the organs. Now, from what I wrote on the board, we should be able to identify systems. Can we do that?
Yes.
Now, you have the nose, which is the example I gave you of the respiratory system. So you have the nose, right? Now, we are not going to be cutting through the brain, but from here, we can actually open this up some more, okay, so that we can project the trachea. Now, can you see?
Yes.
Can you see the trachea?
Yes.
Now I have tampered with an artery, so I'll quickly suck up the blood. And then, of course, you know, I said something about West Being and nobody cared. this thing is supposed to be inside a container, but open it up. You don't like instructions at all. Just keep it inside. So you see, I can't drop this anywhere. It has to be inside my waste bin. All right, so you can see, look at the trachea, right? Can you see the trachea? Now, if you follow this up now, those of you recording, make sure that you are pointing at this particular part. So you can see the trachea. Can you see it? Can you see the trachea? So this is actually projected through the nose, the nostrils here. We follow through here. And then we can follow up the trachea. These are the lungs. Can you see them? You see? This is the left lung. Can you see it? And then this is the right lung. You can see the position of the heart in between them. Okay? You see how the trachea came in. You see the heart. And then you see the splitting of the bronchi. Okay? The bronchi, two of them, bronchus one. So it branched, like this. And then you have the lungs, the right lung, the left lung, and then you have the heart. Now, attached to the trachea is the esophagus, which is the response I tried to give one of your students when she said mouth as one of the organs for respiration. So you see the link. So I'm still having that artery bleeding. So I'll just put my cotton wool there to soak it up. Meanwhile, I'm trying to open up and project the esophagus that is attached with the trachea. Now look at it. Can you see it? Those of you recording here. Now look at the trachea. Can you see it? Attached to the esophagus. Now if you follow up this trachea, if you follow it up, it's behind that way. You now see where it is attached to this guy, which is the what? The stomach. Look at the stomach. Okay. Now look at the esophagus. Look at where it has attached to the stomach. Now the point of attachment is the pyloric, is the cardiac sphincter. Cardiac sphincter, the esophagus comes this way, enter the stomach through the joint here, the muscle called the cardiac sphincter, and then it leaves the stomach through another organ. Look at it. Okay. Now the point at which the muscle that's between the stomach and the exit of the stomach is the pyloric sphincter. So if we are harvesting these organs, we will harvest in such a way that it will show clearly which one is from the cardiac sphincter and which one is from the pyloric sphincter. Now, if you follow this up, you now see that it's the same one that comes through and you have all these convolutions. All right? So this is the colon or the small intestines. Can you see them? Then you have the large intestines. And of course, if you follow the projection, you now see it is actually. Can you see it? Now these are fat bodies. So sometimes if the animal has fat bodies, you may remove some of the fat bodies so that you can see clearly. So we can reduce some of these fat bodies. Okay. Can you see the fat bodies? All right. All right. Can you see the fat bodies? So we just put it. I just want you not to lean. You don't have to lean to see what I'm doing. Don't lean. Okay? And if you follow it up, you will get to the anus. Is that okay? You can see it. Can you see? Can you see it? Can you see the projection? Right? So, we have been able to see the respiratory system, and we also been able to look at the digestive system. So you have the organs. If it is digestive system, like I told you, from the mouth, you have it projected through the esophagus that is attached with the trachea, and then into the stomach, into the small intestines, the large intestines, and then you have the exit, the anus. Is that okay? Now, several other things that you have to see. Now, the urinary system. So let's try to cut. Now look at the urinary system. Also has to do with the kidney. Can you see the two kidneys? Can you see the kidneys? So look at the left kidney. I have the right kidney. Can you see them clearly? Now look at the position. The right kidney is a little bit above the lower kidney. So when you're reading your theory, now you can see, you can connect, you can see it so clear, isn't it? All right. Now, we have, I think we just punctured the bladder. That was what I was sucking away. So we're supposed to be having the bladder somewhere here. So we just punctured it, and maybe it's part of what I remove as the fat bodies. So the bladder is supposed to be projected, and the animal has been starved. Assuming the animal is, you can see even the stomach is small. Ideally, it used to be big, and the bladder will be filled with urine. And since I have punctured it, we can't actually see the projection. So again, the urinary system, you can also start from that point. Now, having done that, we want to attempt to harvest, like I said. So I'm here with chloroform. I'm here with formaldehyde, which is one of our preservatives. We are supposed to have normal saline so that we can prepare formal saline. But let us attempt to harvest for respiratory system and digestive system. So if we are harvesting for the respiratory system, we will try to separate the trachea. Okay, we'll try to separate the trachea from the esophagus. So I can cut it off from this point. Okay, so you can see the trachea, and I'll clearly, and of course this is the liver. Okay, can you see the liver? You can see the liver. This is the liver, and this is the heart. These are the lungs. So I'm going to be harvesting the trachea, the heart, and the lungs. So this is my trachea. So I'll try as much as possible. So you can see I have blood. So it's a small mammal, so that makes it a little bit, where are the forceps? All right, so you can see we have the trachea, but you can see the esophagus still hanging with the trachea, so we can separate them. Can you see the esophagus and the trachea? Can you see them clearly now? Look at the trachea, look at the esophagus. So we try to separate them. Can you see? Can you see? So this is the part that is supposed to go through to the stomach. So we separate them from here. All right. So that if we are doing digestive system, we can follow it up to the stomach where you have the. Can you see? Can you see? Can you see? So this is the cardiac sphincter. So we can go ahead and project our all right. So ladies and gentlemen, this is our respiratory system. Okay. So can you see it? Let me. All right, so look at it clearly. Okay, this is our trachea. Okay, right? And then these are our lungs. You have the right lung, the left lung, and then you have the heart. Now, of course, you would follow through to the part where you have the trachea separating into the different bronchioles. Is that okay? So here we have the respiratory system. So we are not throwing this thing away. We have to fix it and preserve it for subsequent studies. Okay, if you are working on any animal and you want to study the trachea or study the heart or you study the lungs, at this point you have your specimen. So this is what you're going to take for certain studies. Okay, so we'll have containers where we'll prepare the preservative. So for now, we'll just put it by the side. All right. Now, the next thing is the digestive system. Okay. Now, like I said, the digestive system starts from the mouth and then you have the esophagus, then into the stomach. So this is the part that came from the mouth. This is the esophagus, right? This is the esophagus. Now, it is entering into the stomach through the cardiac sphincter. So we're going to be harvesting this linked with the stomach, with the liver, small intestines, large intestines. So that means we are harvesting the whole of this. And you have to be very careful.
Just keep your phone very well. You're obstructing.
All right. So there you go. All right, so we are done. All right, so we have our digestive system here intact. So let me try to arrange it too. So we have the esophagus from up. Okay, I've not detached it. I'll just keep it by the side so that we can look at it. So we have, all right. Look at it. Can you see it? So this is the esophagus. Okay. This is the stomach. This is the liver. So we want to project the pylori and the cardiac sphincter very well. So I need to remove just to separate them here. All right, so this is just what I want to do. So can you see it? So just try to separate. So we reduce these fats. All right, so now we can clearly see the esophagus, okay, joining the stomach through the cardiac sphincter. Look at it there, and then leaving the stomach through the pyloric sphincter. And of course, look at how it goes.
Yeah.
Okay. Now this is called the proximal column. This is the proximal column. Okay, look at it. Now these are the small intestine, and then you have the large intestine. So let's follow through. So from the large intestines, we have projections into the anus. So ladies and gentlemen, for your level, this is the end of this particular session. The idea here is to show you how to open up an animal and then go through the internal anatomy for whatever part you are interested in. As biology students, very soon you get to 400 level where you begin to carry out your respective researches. Those of you who will be interested in working with animals, this is one step into your future work already. So you have this animal, you have this part, whichever one you want to harvest and remove. To the trachea, you have the bronchi, and then you have the lungs with the heart that pumps. The digestive system starts from the mouth through the esophagus into the stomach. It enters the esophagus from the point where you have the cardiac sphincter, leaves the stomach through the pyloric sphincter. You see, cardiac and pyloric sphincters, they are muscle. The first one, that's the reason why you can stand upside down and eat something and swallow and it will come out. So the cardiac sphincters open as you swallow and close. It is only when you have abnormality where the muscle loses its functionality, then it opens up and then you throw up. That's when you see somebody throwing up. Otherwise, that muscle controls that. The same way, once you have this the stomach content in, the pyloric sphincter also controls at what point does the content of the food enters the small intestines. So you have the first point of digestion. Where is the first point of digestion? The mouth. So from the mouth, you have the food eaten being digested. So you have enzymes from the mouth, okay, depending on what substrate or what food you are eating. If it is carbohydrate, you have amylase. that will break down the carbohydrate depending on the different kinds of carbohydrates too. And you also have the ptyalin, okay, from the mouth. Those enzymes are contained in the saliva produced. And then into the stomach, you also have enzymes in the stomach. For example, you have the pepsin, and then you have the renin, you have hydrochloric acid. They are all chemicals that will aid digestion. They are referred to as enzymes. Okay, they will be found here. And of course, you have the liver that will always remove amino acids. They call it deamination of proteins. So the liver, everything you eat in this life will have to pass through the liver. The liver will be the one to remove the toxins and allow the food particles to be digested. Now you have absorption taking place at the stomach. You have absorption in the stomach. You also have another reabsorption in the small intestines, and of course you have the sending in of those food particles or waste into the large intestines, and subsequently they will be released. And like I told you, the urinary system will also include the kidney, the bladder, and then you have the urethra, okay, like that. And of course you also have the reproductive system, which is one of the points I made. So we have to identify the animal, whether it is a male or a female. If it is a male, if it is a female, we're going to see that. I think we have now. This is one gonad. This is one of the gonads. We're supposed to see the second one right in here. So those are the two major things that will identify, make us identify the animal as male or female. So you see the gonads here, the testes, okay, here. Any question? This organism as it is, and we can also go ahead to prepare the skeletal system of this organism. So we still have the skeleton intact, and that one also has its own method, completely different. Is that okay? And then we can remove everything that is inside here and use cotton wool, preserve the skin, use cotton wool, put it right in there and suture it back and preserve it and allow the animal to stay. Is that okay? The animal will stay as if it's a live animal. That one is another course on its own that is referred to as taxidermy. If you enter the lab up, you see some animals that we prepared like that and preserve. So that we can maintain the external anatomy. When you come, instead of killing an animal to study the external, you just come and see it like that. So we expect that we can easily do that, but we don't really have all the materials required to do that taxidermy. We would have removed all these parts and just stuff it up with cotton wool and preserve and allow it to set. If there is no question, ladies and gentlemen, this is the end of dissection using a representative animal.